I'm going to speak about ME/CFS and to some extent long COVID. And what I'm going to focus on are two molecules that are present that have not been studied very much. One is called BH4, and it's an oxidizing reducing agent that's involved in many reactions, and nitric oxide, which is a general regulator of many, many processes in the human body. Partly where this investigation started was the discovery, accidental discovery, by Hector Bonilla that a patient came in to his clinic and said Abilify was helping her. So this is initiated by a patient. Then he tried it working with my research associate, Laurel Crosby, on approximately 100 other patients. And then they reported the response that they got. This is not a double-blind study, unfortunately, because we didn't have the funding to be able to do that. But in this study, we found that around 75% of the patients showed improvement. Some of these are very dramatic improvements. I tried it on my son, and it was very clearly had a very, very strong positive effect. And he was able to do a lot more than he was before. So this set the stage for trying to investigate what happened with the Abilify. So this is the pathways that dopamine is produced. It's thought that Abilify increases dopamine. So dopamine is made from phenylalanine, which is an amino acid that you get in your diet. And phenylalanine carries out a reaction, the hydroxylation reaction, to make phenylalanine into tyrosine. And you can see those are very similar structures. It's an oxidation reaction, but it requires BH4. And then tyrosine is also oxidized to DOPA. This also requires BH4 and to carry out that reaction. And then just as a side note here, tryptophan is converted to hydroxytryptophan, which is the precursor to serotonin. So that requires BH4 as well. So BH4 is involved in production of a lot of the hormones, which could cause a lot of behavioral changes in the patients. Then, to sort this out, we did an experiment with my son where he gave him a bolus of phenylalanine before he ate to look at its conversion. And we took blood samples. He has a Hickman port with time and looked at the level of phenylalanine going to tyrosine and then to other side products. And we found that phenylalanine was slowly converted to tyrosine, and tyrosine was slow to decrease. And so that says it's likely BH4 prey. And that sets the stage for us to explore BH4. BH4 is involved in another very important reaction as well, and that is that arginine is used to produce nitric oxide over here in the circle with NO. So it's just nitrogen combined with oxygen. Very simple gas. It diffuses through membranes. It diffuses very rapidly through the body, and it's used in a lot of things. And maybe people have known that it is involved in dilation of blood vessels. And in the process, it's converted to citrulline. Now, BH4 deficiency is an old idea. It was never really explored very much, and partly because it's very hard to measure. When you do a blood draw, 1% of it's lost from oxidation per minute after the blood draw. It has to be stabilized very quickly. We have now made a contract with Becton Dickinson to put the stabilizers into the blood tubes, and they're a special blood tube So when the blood is drawn, it's automatically stabilized, and that has helped us in making the measurements. Now, when BH4 reacts, it's converted to something called BH2, and then it has to get reduced back to BH4. So that's an oxidation reaction. Well, we've worked out where we can now see both BH2 and BH4 using a very sensitive detector for an HPLC instrument. And what we have found from that is that BH4 is low in the patients. Also learned in this is that the BH2 is actually quite high. So the total amount of BH2 plus BH4 is not necessarily low. In fact, in some of the patients, it's actually fairly high, seemingly that maybe the body is trying to stabilize the production of BH4 by making more substrate for BH4. Also thought that maybe bacteria can make BH4, and that's a very interesting possibility. So it might be for a treatment. And there's some patients are making yogurt with a specific lactobacillus, which I've shown on the screen. But looking at the sequence of the genome sequence of this bacteria, it doesn't have the enzymes necessary for making BH4. However, BH4 is very similar to folate, and a lot of the precursors to folate synthesis are in common with BH4. So it's possible that if you have a deficiency in one of those early steps, it could actually make BH4 from those precursors, and we're exploring that. Also, that this bacteria can be obtained from Amazon, so it's readily available to patients. And some patients feel much better taking it, others do not. Now, the other important component is nitric oxide, which, reminding you, it does also require BH4 to make. And nitric oxide, this is just one of the reactions of many. Nitric oxide is required to react to a metal transporter to be able to transport into cells iron, manganese, and copper. And those metals are often found to be very low in the hair of patients, and hair comes from cells. What's also significant here is those three metals are involved in the scavenging of, as part of an enzyme, part of different enzymes that scavenge reactive oxygens. So this sets the stage that the cells may not have a good ability to neutralize reactive oxygen species. And that could be a very big, important problem. But I also would like to, just as a little bit of a side, that another hypothesis that we have is involved in something called the atacanate shunt. And this is actually fairly new in the literature in terms of even its existence, and most people don't really know about it. So this is laid on top of the citric acid cycle. The citric acid cycle is where most of the ATP is produced. What happens in this reaction is that, and the citric acid cycle is shown you in green, that's the standard cycle. But at cisaconate, there's an enzyme that's produced when you activate innate immunity, and that would be an infection, or even other kinds of things that activate innate immunity. It converts cisaconate. That enzyme goes to the mitochondria, and it converts cisaconate to atacanate. And then atacanate gets combined with coenzyme A, and eventually, it appears to be somewhat slow, it's converted back to pyruvate where you start. So that really robs a lot of the citric acid cycle intermediates that's not being able to produce ATP. And that will generally cause fatigue. And this is something that happens very early when you get an infection and you feel tired. It's probably because of this pathway being activated. So what we've been looking for is a big collaboration with a group in Utah to find a drug that will inhibit cis-aconitate decarboxylase to block the atacanate shunt. And if that's successful, it should generate more energy. They've also set up a zebrafish model where we can do experimentations. They've already identified that cis-aconitate decarboxylate is produced in the zebrafish, and it makes them swim slow. So it looks like this might be a very good model for this disease. Well, one thing I'd like to talk about is, why does this disease last so long? Why do people not get over it? A lot of these things you'd think would reset. You think is the case that this disease is what I would call a cell-autonomous disease, in the sense that individual cells are affected by this, but not necessarily every cell. And if that's the case, then the more cells that are involved, the sicker the patient. It's also likely that the energy depletion, which happens with this disease, and the cells that are affected, it can cause a lot of the systems that you see, and that we think the energy depletion from exertion causes people to crash, just a term that patients have used. It's also possible that the crashes activate innate immunity and starts the disease all over again. This was a hypothesis that was proposed by a graduate student in my lab who came down with ME/CFS, and she worked very hard to not crash, and she did not crash for a whole year, and she got over the disease completely. Well, this is just one example of someone who got cured. We don't know if something else happened. But she's now, the question is she really cured? And she says she runs around 16 kilometers most days with no detrimental effect. The other thing you see in patients is autoantibodies, and this is just a study on one class of those autoantibodies. And this came about by seeing a lot of patients that were diagnosed with MS and then decided that they didn't have MS, they had ME/CFS, and vice versa. Some patients were diagnosed with ME/CFS, found that they had MS. And in talking to some of the physicians, they felt you couldn't get two diseases at the same time. And I think that's false. So what we looked at is studies with the myelin basic protein, and we found that about half of the patients make an antibody that can cleave the myelin sheath basic protein, and that's shown you here. The myelin basic protein is a single band on the gel. When we look at it with ME/CFS patients, we see degraded bands in the gels, and you do not see that in healthy patients. Confirm that this is probably very specific. We looked at a lot of other proteins, and those proteins were not cleaved. Only the myelin basic protein was cleaved by these antibodies.
So that is, we have one last minute.
Okay, so this has appeared that it is cleaving. So the last thing I'd like to show you is this is long COVID patients. These samples came from Sweden, and what we looked at is that we did a virus panel of 20 different, looking at 20 different viruses simultaneously in a multiplex assay. These are almost herpes viruses. What we found is active virus present in the COVID patients, about 80% of active virus. It's EBV. There are other herpes viruses that are also activated, but the EBV is by far the most common. Second one is HHV6, and it's interesting to note that you get EBV or HHV6 or some other virus you don't generally get two at the same time. And after the patients are discharged from the ICU, most of the virus goes away. And that is...
François, I'm afraid we need to stop here.
Okay.
Apologies to you, Ron, and to our listeners for any technical difficulties. But most of all, thank you so much for this presentation and for your ongoing, very, very important work.
Thank you.
Thank you.